Welcome everyone to our first lecture on viruses. Today we will be talking about infectious diseases like rabies and Ebola and the flu and even SARS-CoV-2, that coronavirus that causes COVID-19. But before we can talk about those diseases, we first need to ask, what is a virus exactly? And so a virus is a piece of genetic material that is infectious and it is inside a protein coat. Okay, so that's what a virus is. What is a virus made of? If you haven't yet, go ahead and paste this diagram into your notes. You should have it in your STT kit. So cut it out, paste it in. We are going to label it. First thing to see on our virus, the squiggly stuff in the middle is the genetic material. And I say genetic material because it could be RNA or it could be DNA. It depends on the virus. Now, typically, RNA has a single strand. And DNA has double strands. It's a double helix. Viruses are unique because some viruses actually have double-stranded RNA or single-stranded DNA. Now, a virus reproduces by taking over a host cell, and most of these host cells do not have the means to replicate RNA. They only have ways to make more copies of DNA. So a retrovirus is a virus that has RNA inside of it, okay? The flu is an example of a retrovirus. Now, when this RNA gets inside the cell can't copy it itself. And the RNA has to be reverse transcribed to DNA. And the enzyme in a retrovirus that does this is called reverse transcriptase. So that is the enzyme that reverse transcribes RNA to DNA so that the host cell will then have DNA to copy to make more RNA. Okay? So that's the genetic material inside a virus. Around the genetic material is the capsid, which is the protein coat. And it often looks a little bit crystalline, like it does in this picture. Okay. Around the capsid, a virus may or may not have an envelope. And that envelope is, if you look closely, a phospholipid bilayer or a membrane. And it is either stolen from the host's outer membrane or from the host's Golgi or ER. Okay, so that membrane is stolen. In the envelope, or attached to the envelope and sometimes attached to the capsid, there are glycoprotein spikes. And if we look at this word glycoprotein, glyco just means sugar. So these are protein sugar complexes. I'm not sure why my pen isn't writing. Okay. And even though they're called spikes, they're not really spiky or pointy. They're not used for defense. They are used for attaching to the host cell. If you've seen a picture of coronavirus, the little red things on the outside, those are the glycoprotein spikes. Okay, so that's what's inside our virus. Now, I want you to take out this Venn diagram that you worked on as your opener, and we are going to check our answers and fill in, and we're going to add viruses to it since you hadn't looked at viruses before you started doing that activity. So let's first talk about the features or organelles that are in all three. Okay, so we're looking for the presence of these things, and they are structures, not characteristics. So viruses, prokaryotes, and eukaryotes all have proteins. They all have membrane. And they all have some kind of genetic material. Okay, if we think about what only viruses have, though, only viruses have that capsid, the protein coat. Only viruses have glycoprotein spikes, although other cells do have surface proteins. And only viruses have single-stranded DNA or double-stranded RNA, but not all viruses have those. Okay. Now let's think about what do prokaryotes and eukaryotes both have that viruses do not. Okay. Prokaryotes and eukaryotes both have ribosomes. And I'm going to make this section a little bit bigger just because this is a big section. So they have ribosomes. They have cytoplasm. They have a cytoskeleton. And they have metabolic enzymes, meaning they have ways to process energy and create ATP. Okay. If we then think about what only bacteria have that capsule on the outside, the polysaccharide and sometimes lipid layer around the cell wall. Okay. Only bacteria have pili, and only bacteria have a plasmid. Now, the reason I'm not putting cell walls on here is there are some eukaryotes with a cell wall and some without. And so that's why that one's not quite on there, because it's not a universal characteristic. Our eukaryotes, though, they all do have a nucleus, and they all have membrane-bound organelles. If we think about what viruses and eukaryotes have in common, there really isn't anything they have in common that bacteria don't have. And the same with viruses and prokaryotes. Anything that they have in common, they also have in common with eukaryotes. And it all goes in the middle right here. If we think about these, which of these structures are the reasons that viruses need a host to reproduce? And the reason that viruses need a host to reproduce is because they do not have ribosomes to make proteins, and they do not have metabolic enzymes, okay? Remember that proteins are what do all the jobs in the cell. So if you don't have proteins, you can't do any jobs. You can't make any more of yourself, okay? So viruses need a host in order to take their ribosomes and make proteins, but also viruses have no way to make ATP because they have no metabolic enzymes. And so they have to take over a host cell and use the host cell's metabolic enzymes or mitochondria to make energy. So these are the two reasons why viruses cannot reproduce on their own. So we didn't just use those M's, L's, and N's for fun. We are going to use them so that you can self-assess how much you remembered about cell structures before and how much you still needed assistance with learning. So what I want you to do is add up the organelles you correctly placed using each of the following strategies. How many M's did you get correct? How many L's were correct? How many N's were correct? And how many had no letter, meaning we corrected them during the lecture? Okay. So pause the video, add them up, and write those totals in the margins of your notes right next to your diagram. You're then going to use those totals to figure out your study strategy for tonight. If you correctly placed more than half of the vocab words with no help, meaning that you had 7 or more M's on your paper, then your study strategy tonight is to review your notes. If you correctly placed more than half of the vocab words using the word bank, you need to, then that means if the sum of your M's and L's together was more than 7, then you need to make flashcards for each of the cell types. If you placed more than half of the vocab words using notes, meaning that you had to use your M's, L's, and N's added up, that's more than 7, then you need to make flashcards for each of the cell types and highlight your notes. And then if you correctly placed less than half of the vocab words during the opener, and you had to correct most of them during the lecture, meaning that your M plus L plus N was less than 7, you need to schedule a tutorial or come to office hours tomorrow morning so that we can talk about some strategies for you. And you may also want to try making some flashcards and highlighting your notes tonight as well. Make sure you write your study strategy in the margin of your notes, which one you are going to do tonight based on your score. So let's pause for a moment and take a look at virus diversity. Viruses are very diverse, and viruses may or may not have all of the structures that we saw on the previous slide. So some viruses are just a capsid plus the genetic material. Some have the capsid, the genetic material, and glycoprotein spikes. Some have multiple capsids with multiple pieces of genetic material and the envelope with glycoprotein spikes. And then this one, the bacteriophage, is a very special bacteria, or is a very special virus that infects bacteria. And in my opinion, it looks like an alien landing pod. Okay. If we then take a look at coronavirus in particular, coronavirus has RNA inside of it, okay? That is its genetic material, and surrounding the RNA is the nucleocapsid. And then outside of that is the envelope, okay, with the glycoprotein spikes and membrane proteins in it. So that's our coronavirus structures. So now that we know what a virus is made of, we need to know how does a virus infect a host. And we're going to watch a quick video on that to see how it happens.
Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core, RNA, and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface where they combine with the encapsulated nucleoproteins to form progeny viruses which depart from the cell by budding. The virion now goes on to infect other cells. At present, vaccines to stimulate the production of antibodies against the virus and stimulate host cellular responses to ingest the virus remain the best strategy for controlling certain viral diseases. But they are of limited effectiveness against rapidly mutating viruses and are ineffective against viruses that cause the common cold.
So to get a more in-depth look at how a virus reproduces, we're now going to do a virtual lab and simulation. Your notes are not done for the day. You will take more notes during live session, but you can set aside your notebook for right now. So to do today's virtual lab or simulation, you're going to go to Google Classroom and complete Day 4, Microbiology, Lab 4, Virus Lytic Cycle Exploration. And then come to live session ready to talk about what you've learned.